Dear students, in this module we're going to complete the flowchart for the Chou-Fasman algorithm towards predicting the secondary structures using amino acid sequences of proteins. You know that the Chou-Fasman algorithm predicts the alpha helices, the beta sheets, and the beta turns as well. So I'm going to review the beta sheets and the alpha helices and their flowcharts. So as you already know that you scan the entire amino acid sequence as shown here, and you see if 4 out of 6 amino acids do not have a propensity for forming an alpha helix. So if the propensity for forming an alpha helix is low, then you check for their propensity for forming a beta sheet. So if the propensity for forming a beta sheet is good, then you move forward towards forming a beta sheet. But in case if the propensity for forming an alpha helix is high, then you must form an alpha helix. So let's review that portion. We extended the flowchart of forming beta sheets to include alpha helices as follows, where the amino acids had a propensity for forming an alpha helix, and we extended the helix like that. During this process, we saw if the average propensity over the entire sequence for forming an alpha helix decreased to below 1, and in that case, we returned to the formation of beta sheets. Now we are going to modify this flowchart again and include the portion for predicting the beta turns. For that, I'm abridging and summarizing the portion, the flowchart portion, which predicted the alpha helices by this yellow box. We are talking to this yellow box if the propensity for forming an alpha helix is high here, or if the average propensity for forming an alpha helix fell below 1. So these are the two interactions between this yellow box and the rest of the flowchart. Now we are going to extend. So if a sequence did not have the propensity for forming an alpha helix and it did not have the propensity for forming a beta sheet as well, so all we are left with is to compute the propensity for these amino acids to form a beta turn. For that, we compute the F total as given by the Chou-Fasman algorithm. So there are 3 conditions after this. So if the F total that you have computed is greater than 0.000075, or the average propensity for the turn is greater than 1, and if the propensity for the turn is greater than both the alpha helix and the beta sheet, in that case you form a turn. So the region is finalized as a beta turn, as I just mentioned. But if that is not the case, then you can also have a region where the propensity for forming a beta sheet was low, and then you may have to compute the F total yet again for that portion. Also, there can be another case wherein your 4 contiguous amino acids do not have a propensity high enough. That is, they are less than 1 for forming a beta sheet. In that case, you also need to compute the F total towards computing the beta turns propensity. So you continue to do that until you finalize the entire sequence, in which case you finish the prediction algorithm here. But if you still have amino acids, then you can continue to scan the sequence and finalize beta turns, beta sheets, and alpha helices like that. So in conclusion, as you just constructed the flowchart for predicting the alpha helices, beta sheets, and turns, you can see how the Chou-Fasman algorithm relies on the statistics that we derive from the databases. And those statistics, they tell you about the occurrence of specific amino acids in certain portions of the secondary structures. Now we will evaluate how these algorithms perform in the later modules.